Absorbance and fluorescence. Some background information is that absorbance is the measured amount of light that can be absorbed from the sample in the cuvette. By using spectroscopy, a measure of how much light is absorbed in a given path length can be determined. Spectroscopy takes the sample and shines the light through, causing a dispersion of energy levels to be emitted. Fluorescence is the process of molecules having their electron states excited and produces light as the molecule returns to lower energy states. The research question is by using the absorbance fingerprint of our solution, what is the kind and concentration of dye in the sample? And also, what is the absorption and emission spectrum of the sample of a single fluorescent dye? Some assumptions that we had were prior to any data collection, the dye gave an appearance of a bluish color, which allowed the assumption of Lapidus dye. And we assumed our dye had a molar substance characteristics consistent with those given in class.
To help identify an unknown sample, absorbance can be used to measure the concentration of a molecule in solution or find what type of substance it is. Spectroscopists can identify an unknown sample using the absorbance spectra because spectroscopy measures the absorption of radiation of light in that sample, along with the frequency and wavelength of the light that shines through the sample. Based off of the wavelength and corresponding frequencies of light, they can match the sample with other known absorption spectra, which is what we perform as a group in this experiment. Beer's law is also helpful with finding the concentration of a substance because the amount of light absorbed is proportional to the concentration of that solution. So using the Beer's law's equation, you can measure how much light is absorbed in a given path length because of the properties of the material through which the light is shining and traveling through.
There are key differences between absorption spectra and fluorescence spectra. Absorption spectra involves measuring the absorbance of a sample at particular wavelengths of light based on photons' interaction with a sample. This is useful in determining the concentration of a compound. This is done by understanding the principle that more light absorbed means there's a higher concentration of the compound, and Beer's law can be used to determine quantitative data. On the other hand, fluorescence spectra involves measuring the fluorescence of a compound at particular wavelengths of light through excitation of electrons. This is useful in analyzing compounds in the medical and biochemical field, such as tracking movement of a compound after it fluoresces. A critical difference between absorption and fluorescence spectra is that the wavelength reaching the detector in fluorescence spectra is a different wavelength than the initial light. It is emitted at a higher wavelength and lower energy than its original input. Our experimental setup involved recording spectra of light coming through a 1 centimeter cross-section cuvette containing dye solutions. Data was obtained through a software program attached to the fiber optic cable, giving live readings back to the computer. We found it was imperative to prop up the fiber optic sensor as it had a tendency to tilt down and obtain insufficient data. Recording data in a dark environment is necessary so the sensor picks up minimal noise from other external light sources, such as the overhead lights in the classroom. The light source was placed 90 degrees from the sensor to avoid interference of the transmitted excitation light and obtain a more pure reading.
From the unknown dye, we observed peaks at 550 nanometers and 600 nanometers. The Lapidus dye showed peaks at 550 nanometers, while the Sautel dye showed peaks at 500 nanometers. Given that the 600 nanometer spike could occur from surface interference on the cuvette, we concluded that our dye was the Lapidus dye. We then applied the absorbance equation of Beer's law to calculate the concentration given the coefficient at 550 nanometers by dividing our absorbance value by the coefficient for the Lapidus dye and by 1 centimeter. This resulted in 55.2 micromolar, or 64.2 micromolar if the 600 nanometer peak was used instead. From our data from the fluorescence, we observed that the absorbance was around 495 nanometers and the emittance was around 520 nanometers to 522 nanometers. The slight negative value that we observed was likely due to the higher absorbance in water than in the dye. As seen in the fluorescence and molecules activity, molecules undergo simple harmonic oscillation and can absorb photons within a range of wavelengths. They favor high-frequency wavelengths for raising to an excited state, then nuclear position shifts and the emitted photon is of lower frequency. Thus, energy is ultimately conserved due to the movement of the nuclei utilizing the remaining energy from the excitant photon.
Thanks for watching.